Okay, so very good afternoon to all. Today we are going to start a new topic in biochemistry, namely enzymes. Yes, so we had discussed about the basic aspects of biochemistry, that is pH and buffer, and also we have seen introduction to carbohydrate, protein, fat, their classification. Yes, before going to metabolism, I thought why don't we go for application of both pH as well as protein aspect that we had discussed, hence the term enzymes. So in proteins also we had discussed this thing that enzymes are mainly made up of proteins or group or collection of amino acids. So the same thing we are going to discuss in detail. Yeah. So this will be the main content. First will be the chemistry that we are going to see what are they made up of, as we already know amino acids and proteins. What is the structure we'll discuss. Then we'll see about their classification, and last will be the mechanism of enzyme action and the effect of pH, temperature and other sources on the enzyme action. Yeah. So first, going for chemistry. Enzymes are biological catalysts that speed up the rate of biological reaction. So what is the function of enzyme? Enzyme has nothing important function if you see from substrate and product. Say for example, A plus B equals to C. Yes, that reaction takes place in 5 minutes. To speed up this reaction, whatever is required is termed as enzyme. So A plus B equals to C in 5 minutes is without any enzyme, but the same reaction can be faster. એક જ reaction ને fast કરવું હોય, biochemical reaction ne fast કરવું હોય, that means we have to use an enzyme. Enzymes are naturally present in the body. We do not have to provide it from external sources. Most enzymes are three-dimensional globular proteins. We discussed about the types of proteins. Yes, one of them was globular. So globular protein which has tertiary and quaternary structures. Again, primary, secondary, tertiary, quaternary. Primary was single chain. Yes, collection or arrangement of amino acids. There are mainly 20 basic types of amino acids present in the body naturally. Yes, એના જ combination થી different type of proteins બને છે. એ એક ચેનમાં થાય એવી ઘણી બધી ચેન્સ થાય, that is secondary. These chains may be in the helical structure, alpha helix. Yes, remember the terms alpha helix. They together form a tertiary structure which is 3D, and such simple molecules combine together to form quaternary structures. Yes, the same thing, all things we have discussed in protein. That is why I took the topic, so that it will be revision of proteins, as well as we can move forward and learn about enzymes because they are made up of proteins. So tertiary, quaternary structure, these are globular proteins. Some special RNA species also act as enzymes and are called ribozymes. Example, hammerhead ribozyme. So as the name suggests, hammerhead. Yes, it is its head or the part which is going to be working as enzyme will be in the form of hammer. Yes, what is a hammer? What do you call hammer in Gujarati?
Authority.
What is hammer? Very you understood the shape. The shape is like a square or a rectangle and followed by a single strand pipe. Yeah, so that is hammerhead ribozyme. So mainly you have to remember they are biological catalyst. Catalyst means they will help in the process, further process કરાવશે. Second, they are globular proteins having tertiary and quaternary structures. Here we have seen collection of alpha helix and beta sheet. Yes, remember those terms. And there are some special RNAs known as ribozyme which also act as enzymes. Structure. So the active site of an enzyme is the region that binds substrate, cofactors and prosthetic groups and contains residue that helps to hold the substrate. Now enzymes have specific region. Enzyme નું કામ શું છે કંઈક પણ process ને specifically fast કરવી. Yes, so for that part they have a specific site. Site એટલે કે કેવી જગ્યા છે enzyme ના structure માં જેને કહેવાશે active site. Here what can attach? એ એક region છે જ્યાં ઘણું બધું attach થઈ શકે. So first is the substrate. જે A plus B equals to C છે તો A plus B એ બંનેમાંથી કોઈ પણ એક ક્યાં join થશે, that is termed as substrate. Cofactors, some other factors which can help in progressing of the reaction. Yes, that is cofactors and other groups, prosthetic groups, that is some extra types of groups can also attach to the enzyme and they can move forward the reaction more rapidly. Active site generally occupy 5% of the total surface area of enzyme. you consider a complete big balloon. Yes, એનું ખાલી 5% area જ active site હશે, બાકી 95% will be the specific shape of that enzyme. Yes, the balloon that we consider, that is, it is circular. Yes, circular portion is responsible to attract various things. એ circular હશે તો જ અમુક ટાઈપના સબસ્ટ્રેટ એની પાસે આવશે, બાકી નહીં આવે. Yes, so shape is 95%, 5% is only the active region. એ active region માં જ બધું bind થશે. Active site has a specific shape due to the tertiary structure of protein. As we already know, tertiary structure, that is 3D model. Yes, so it is a 3D structure. So it has specific, very specific shape also. All enzymes have specific shapes. A change in the shape of protein affects the shape of the active site and function of the enzyme. Now, as we described, એક balloon છે, એની જો rectangle shape નું થઈ જાય, active site પણ change થઈ જશે. જો active site change થઈ ગઈ એટલે substrate જે પહેલા attach થતા હતા એ નહીં થાય. Yes, so there will be new substrate and the enzyme will help in some other type of reaction. Yes. So shape of the enzyme is utmost important and active site is important. The whole shape of the protein or enzyme will determine the shape of the active site. Active site depend કરશે આ પ્રોટીન આખા એન્ઝાઇમ ના સ્ટ્રક્ચર ઉપર. જો સ્ટ્રક્ચર ચેન્જ થઈ જાય તો એક્ટિવ સાઇટ પણ ચેન્જ થઈ જાય. એક્ટિવ સાઇટ ચેન્જ થઈ જશે, that means different type of enzymes or different type of substrate will combine and will move forward the reaction. So active site, it has two important functions. Active site can be divided into binding site, catalytic site. Binding site, it chooses substrate and binds with active site. Catalytic, it performs the catalytic action of the enzyme. Substrate directly આવીને, say for example, leads. Yes, you want to make juice from a sugar. Yes, તો એને શું જરૂરી છે? You need to have a machine that can grind the sugar and develop a sugarcane juice. So A is directly C. Clear? A, that is sugar, sugarcane. C, sugarcane. So binding substrate bind એટલે sugar directly say that to of the is the can juice, but forms the say some amount. Yes, some lemon will go form sugar which is heavy. That so that is a binding direct say શું કરશે અને is site two function depending on the nature. It site important here. It does have an what are molecule વસ્તુ નથી. It may be organic, it may be inorganic. Inorganic as in form of any mineral. Yes, which carries out reaction cannot perform. We know 20 acids.
Sir, your voice is breaking.
Protein. Yes.
Sir, your voice is breaking, so we cannot understand what you are saying right now.
Okay, I guess connection went off. Able to hear me?
Yes, sir.
Okay,
Yes,
able to see the presentation?
Yes,
yes,
sir. Yes, sir.
Okay. So, are you clear? Did you hear about the active site, the examples of binding and catalytic site? Well, is it clear?
No, sir. Can you repeat it?
Okay, sure, sure. Fine. We'll start from the structure of the enzyme. Yes. So the structure of the enzyme, it has an active site. Active site means એક એવી જગ્યા આખા enzyme which is only 5%, a small region જ્યાં substrate or other factors that we talk about, cofactors that we are going to see after this, they attach themselves. After they attach themselves, the enzyme will be able to produce its action. May that be in the form of producing a product or rapiding the or increasing the pace or fasten the process. Yes. So active site occupies 5% of the total surface area of the enzyme. Active site બહુ જ નાની જગ્યા છે. Yes, the example that we took, a big balloon. Yes, so it will be round in shape, but only 5%, the top region or specifically any part. We take example of top region where other things can attach. Say for example a string. Yes, so that is 5% total surface area of the enzyme is only active. Active site is very specific. So active site જે shape નું હશે, you might have heard the term lock and key mechanism. Yes, so a lock can only be opened when there is a specific key attached to it, otherwise it will not open. The same thing is for active site. Active site is like a lock. Yes, and the substrate will be the key. Whenever lock and key mechanism or lock and key match in their combination, then only the process will move further. Otherwise it will not move. So this lock as a whole will depend, its shape will depend. Yes, hence a change in of the protein affect the shape of the active site and function of the enzyme. So whenever you see the function of the enzyme is changed, more or less it will be because of the change in shape of the protein. Active site ક્યારે active થશે? જ્યારે exact એના opposite type નું substrate એની પાસે આવશે. Yes, so this its shape depends, or active site activation depends on shape of the protein. Moving to active site. Now we talked about active site. Active site na બે function છે, binding and catalytic. Binding means A product directly gives B. Yes, A leads to B. A, say for example, we take example of sugar. So a sugar cane can be converted to sugar cane juice. Yes, when you have a machine. So this machine is an enzyme. Yeah. So A directly binds with active site. Sugar cane directly goes into the machine to produce sugar cane juice. That is binding site. If you want to also add lemon, so A plus B leads to C. So sugarcane plus lemon in the machine, that will lead to production of citrus or citric sugarcane juice. Yes, it will be somewhat citrus. So A plus B leads to C. So this is catalysis. It will only aid in the action. Two substrates are there which are producing a product. That will be catalytic action. Catalytic action means catalysis. Catalysis means it fastens the process. E process ne jaldi karavse. There is no other change. Yes, but binding site, it is required, then only product will be formed. So A cannot directly lead to B without the active site, the binding site. Yes, so that is binding site and catalytic site. Now we talked about Now we talk about the structure of the enzyme. So in that we saw about the substrate. Substrate means something that will react to form a product. Yes. Next is cofactor. So what are cofactor? Cofactor is nothing but a non-protein molecule. Maybe carbohydrate, it may be fat, it may be some ions, it may be minerals, anything else. Yes, which can carry out a chemical reaction which cannot be performed by standard 20 amino acid. Now we know in our body we have learned there are specifically 20 amino acid through which there is formation of protein, enzymes, etc. Yes, but what if a substrate requires some other thing to be attached? Yes, ions, say for example, non-inorganic cofactors like ions are also needed for reaction. So here we need additional cover. You can see here the cover. Yes, so this is a protein. You can see the shape of the protein. Yes, very specific. If the whole structure of the protein is changing, this of active site will also change. Yeah, so this cofactor, co it will be along with associated and factor. So it is something that is associated with which helps in reaction or process. So this inactive protein, when cofactor attaches to it, it will have active site, and here there will be substrate binding. Yes, the substrate, that is the main reactant that will form the product, that will come and bind here. You can see these are the active sites. This is the active protein with cofactor, and here will be the binding site. So you can see the shape, yes, completely opposite to the active site of the protein. So this is lock and key mechanism. Yes, there are other theories also that we'll be discussing how how enzyme works. Yes, so enzyme કઈ રીતે કામ કરે છે એ આપણે differently જોશું, but for now lock and key is easy to remember. So you take the example of the same and remember. So again, cofactors are of two types. It may be organic, it may be inorganic. Coming to inorganic cofactors, so these are the inorganic molecules required for the proper activity of enzyme. So these are additional things that will attach to the enzyme which are non-proteinous. Yes, cofactors are not proteins. Remember this thing. Cofactors are never proteins. If they are proteins, they are enzymes itself. Example, enzyme carbonic anhydrase requires zinc for its activity. Hexokinase has cofactor magnesium. Yes, so these are example. These are ions. They are not proteins. They are ions which have nothing to do with the protein structure. Yes, and they will attach on the enzyme so as to they can fasten the reaction. Also see how an enzyme is being named. You can see carbonic anhydrase, ASE, hexokinase, AAC. Yes, so whichever the substrate is there, તમારો product use substrate use થવાનું છે, જે પણ reactants છે તમારા એ use થવાના છે, એને એના જ નામમાં if you add ASE in that, it will be the name of the enzyme. We are going to see the nomenclature of enzyme, so just a hint from it. Organic cofactor, inorganic. Inorganic, why? They are the ions or minerals that will be responsible for cofactor work. Organic, these are organic molecules required for proper activity of enzymes like glycogen phosphorylase. Yes, ASE again requires the small organic molecule pyridoxal phosphate. So that is a organic compound. Yes, so because of this presence of cofactor, it will be helpful in the process. Otherwise it cannot perform any process. It will be an inactive protein. Yes, જ્યાં સુધી cofactor અહીંયા attach નહીં થાય ત્યાં સુધી inactive હશે. Inactive means active site નથી એની પાસે. Active site નહીં હોય તો substrate join નહીં થાય. If substrate is not joining, there will be no product formation. Yes, coming to term substrate, the reactant in the biochemical reaction is termed as substrate. જે પણ join થવાનું છે, જે react થવાના છે બે product, so you can say Na plus Cl gives NaCl. Yes, so this normal procedure is also happening. So Na and Cl individually are substrate. NaCl that is formed is the product. So substrate એટલે જે initially react કરવાના છે. CO2 plus H2O gives H2CO3. Yes, in the presence of carbonic anhydrase. Yes, so here zinc is needed, as we already discussed in the cofactor, and also CO2 and H2O individually are the substrates. When a substrate binds to an enzyme, it forms an enzyme substrate complex. Easy. Enzyme substrate જ્યારે bind થશે enzyme ને એને કહેવાય enzyme substrate complex. So here substrate will join in the enzyme. Yes, again the shape is like a lock, and this substrate will be the key, then only it can join, otherwise it cannot join. Now coming to sites of enzyme synthesis. Enzymes આપણે આટલી વાત કરી કે protein હોય, બધી process માં help કરે, it can bind directly to substrate and it gives product, but enzymes બને ક્યાં? Yes, so this answer again we have answered in the proteins. Yes, so proteins are formed from DNA. Yes, specific part of DNA termed as gene. એક gene transcript થશે, એ transport RNA tRNA બહાર લઈ જશે અને ribosomes એને decode કરશે અને proteins બનાવશે. Those proteins are nothing else but enzymes. So enzymes are synthesized by ribosomes which are attached to the rough endoplasmic reticulum. Now this you need to remember, go back to histology and in your 11th and 12th you were the cellular structures or the organelles present in a cell. Yes, so mitochondria, ribosome, endoplasmic reticulum. Endoplasmic reticulum are again of two types, rough and smooth. Yes, nucleus. Nucleus contains DNA. So these are all the organelles that are present. They are suspended in the cell cytoplasm. So enzymes are synthesized by ribosome, which are attached to the rough endoplasmic reticulum. Rough and smooth endoplasmic reticulum ma difference એટલો જ હોય કે rough endoplasmic reticulum have ribosomes on them. Yes, એની ઉપર ribosomes હશે, that is where they are termed rough. Information for the synthesis of enzyme is carried by DNA. Yes, parts of DNA. Yes, that we talked about, which is termed as gene. Amino acids are bonded together to form specific enzyme according to DNA's code. So whole DNA is there. From that, a specific part is taken. It is transcripted, that is transported by RNA in the form of RNA, and later on these ribosomes decode and form a specific protein. So this is how a enzyme is synthesized. Enzymes are always, or proteins are always synthesized in ribosomes. Remember this, that are present on rough endoplasmic reticulum, RER, rough endoplasmic reticulum. It is made from our DNA. Yes, specific parts of DNA. Now there are two main types of enzymes, intracellular, extracellular. નામ ઉપરથી જ ખબર પડી જાય, જે સેલની અંદર કામ કરશે that is intracellular, જે સેલની બહાર જઈને કામ કરશે, બીજી જગ્યાએ કામ કરશે, that are extracellular. Intracellular enzymes are synthesized and retained in the cell for use of the cell itself. એટલે કોઈ પણ એન્ઝાઇમ રાઇબોસોમ્સે બનાવ્યું, ઈ જ સેલમાં પ્રોટીન, ઈ જ એન્ઝાઇમ કામ કરે, એને કહેવાય intracellular. They are found in cytoplasm, nucleus, mitochondria, chloroplast. Yes, within the cell જે પણ organelles છે, say for example mitochondria, which we know is the powerhouse of the cell, એનું કામ શું છે? પાવર જનરેટ કરવું. તો ATP માંથી ADP બનાવવા માટે પણ enzymes જોઈએ. Yes, so if a ribosome present on the rough endoplasmic reticulum goes in the mitochondria and helps in this process of conversion of ATP to ADP, that is example of intracellular enzyme. Excuse me. Example, oxidoreductase catalysis biological oxidation. As the name suggests, oxidoreductase, so it will lead to oxidation and reduction of a specific substrate. It catalyses biological oxidation. Enzyme involved in reduction in the mitochondria. Yes, the same example that I gave you for conversion of ATP to ADP. That is role of enzyme, intracellular enzyme, because the enzyme is formed in the same cell, it is being used by the same cell. Extracellular enzymes means it will be produced at one specific region, but its action will be somewhere else. So these are the enzymes synthesized in the cell, but secreted from the cell to work externally or somewhere else in the body. Digestive enzyme produced by the pancreas, yes, are not used by the cells in the pancreas, but they are transported to the duodenum, and where they will form the or do the process of digestion and absorption. Yes, formation of bile. Yes, so these are the examples of intracellular and extracellular. Extracellular એટલે બહાર જઈને produce એક cell કરશે, ઈ enzyme બહાર જશે, બીજી જગ્યાએ action કરશે, that is extracellular. If it does in the within the cell, it is intracellular enzyme. Characteristics, utmost important. Yes, enzyme, કોઈ પણ protein ને enzyme ક્યારે કહેવાય? This is the main characteristic of enzyme. So enzymes speed up the reaction by lowering the activation energy of the reaction. Yes, say for example CO2 plus H2O leads to H2CO3. This process will happen even in the absence of carbonic anhydrase. Carbonic anhydrase nahi hoy to પણ એ reaction થશે, but that reaction will be slow, and to initiate the reaction the amount of CO2 and H2O required will be very high. કાર્બન ડાયોક્સાઇડ અને વોટર બહુ વધારે પ્રમાણમાં જોશે, પછી આ એક્શન સ્ટાર્ટ થશે, રિએક્શન સ્ટાર્ટ થશે. But what is the function of enzyme? It will speed up the reaction by lowering the activation energy of the reaction. જો energy required હોય, જે substrates required હોય ઓછા હશે તો પણ ઈ action કરાવશે, તો પણ ઈ reaction fast કરશે. Yes, so that is the first thing, reaction, speed up the speed of reaction. Second, their presence does not affect the nature and properties of end product. Yes, substrate માં એ changes કરશે, enzyme bind, substrate bind થશે enzyme માં, તો substrate ની properties change થશે, પણ it will never change the product. Yes, so CO2 plus H2O એ bind થશે enzyme ને, carbonic anhydrase ને, એટલે CO2 અને H2O ની properties change થઈ, પણ end product શું થયું? H2CO3, ઈ એની properties change નહીં કરે, never. So this does not affect the nature and properties of the end product. Substrate to change થવાનું જ છે. Why? It is attaching to the enzymes. So any properties change થશે, physical and chemical. But it will not change the properties of end product. They are highly specific in their action. That is, each enzyme can catalyze one kind of substrate. Yes, so H2O plus gives H2CO3. Carbonic anhydrase is the enzyme. Carbonic anhydrase can only be used for this conversion of CO2 plus H2, H2CO3 or H2CO3 into CO2 plus H2O. It cannot work anywhere else. NaHCO3 plus H2O करवा जाओ तो ये carbonic anhydrase તમે use करो तो there is no reaction. It will not help in that reaction. Why? It is very specific. ये एक જ type ना substrate ने product માં convert करશે. That is highly specific. Small amount of enzymes can accelerate the chemical reaction. Yes, as we talked about, substrate ઓછા હશે તો પણ એ product બનાવશે. Yes, ideally એવું ના થાય because you need large amount of substrate, then only it can form a product. But because of enzyme, even small amount of substrate is there, it can lead to product formation. Enzymes are sensitive to change in pH, temperature and substrate concentration. ત્રણ વસ્તુ enzyme ની activity change કરે. pH, temperature and substrate concentration. How they will change, we'll see in the further classes. Yes, so just for now, remember three things are utmost important. First is pH. Yes, that is why our human body requires to be maintained at specific pH and temperature. Yes, specific pH હશે અને specific temperature હશે તો કામ કરી શકે. જરાક પણ temperature alter થશે plus or minus. Yes, or pH, if it increases or decreases, acidosis or alkalosis, what we have discussed in the pH and buffers, if anything as such happens, then enzymes will not be activated, your biological processes will stop, the metabolism of the body will stop, and if it stops, it is life-threatening condition. Yes. And last part, turnover number is defined as the number of substrate molecules transformed per minute by one enzyme molecule. So you need to remember this unit, turnover number. એટલે કે કેટલા substrate એક મિનિટમાં ટ્રાન્સફર થઈને પ્રોડક્ટ બને છે with the use of one enzyme. એક enzyme per minute કેટલા substrate ને પ્રોડક્ટમાં કન્વર્ટ કરે છે, that is termed as turnover number. Catalysis part, what we talked about two, binding and catalysis action. Yes, action માં આપણે જોયું active site માં. So for catalysis, turnover number is 6 into 10 raised to 6 per minute. Yes, 6 into 10 raised to 6, 6 followed by 6 zeros. Yes, આટલા substrate per minute એક enzyme molecule ના લીધે product માં convert થાય. Yes, so this is the main indication, or you can say main action of enzymes. It converts the substrate so fast into the product. Yes, so it speeds up the thing. The main characteristic, it speeds up the reaction. It does not affect properties of the end product. It is highly specific. Even if the substrate are lower in amount, then also enzyme acts. They are very sensitive to change in pH, temperature and substrate concentration. And last is the turnover number. 6 important characteristics. This can be asked itself as a 5-marker question. Yes. Nomenclature. Yes, how do you name enzymes? Enzymes na naam કઈ રીતે પાડવાના? So it is simple, as we talked about. Substrate નું જે હોય નામ, એમાં ASE પાછળ લગાડી દો, that is an enzyme name. Simple as that. Yes. So enzyme is named according to the name of the substrate it catalyzes. જે substrate હશે એના નામ ઉપરથી જ enzyme નું નામ પડે. Some enzymes were named before a systemic way of naming enzymes were formed. So example, pepsin, trypsin, renin. All the three enzymes, they are responsible for digestion process. You have come across them in physiology as well as in your 11th and 12th. Yes, so pepsin, trypsin and renin, all the three are responsible for digestion. So before this nomenclature came, પેલા જે આપણે decide કર્યું કે substrate ઉપરથી enzyme ના નામ પાડશું, એ nomenclature આવી, system આવી, એની પહેલા જ these were named. Yes, hence these are also enzymes, pepsin, trypsin, but they do not have ASE at the end because before the nomenclature came, they were already named. એમને નામ પહેલા જ આપી દીધું હતું જ્યાં સુધી nomenclature system આવે. By adding the suffix ASE at the end of the name of the substrate, enzymes are named. Simple as that. ASE at the end of the substrate, name of the substrate. Example, maltose plus water. So maltose, OSE ne કાઢીને ASE, maltase leads to glucose plus glucose. Giving few examples, remember 2 or 3 if possible. It is easier to remember compared to other things. So lactose, so lactase, maltose, maltase, cellulose, cellulase, lipids, lipase, starch, amylase, protein, protease. Yes, so these are the examples. And if you want to know the product also, if lactose is given enzyme lactase, it will lead to glucose plus galactose. Maltose will form 2 molecules of glucose in presence of maltase. Cellulose will give glucose, again multiple molecules of glucose, because cellulose is a polysaccharide. Poly means many molecules, saccharide, yes, carbohydrate, as we had discussed. Lipids in the presence of lipase will give glycerol and 3 fatty acids that we had discussed. OH group of glycerol, 3 OH group in glycerol will attach with 1,1 fatty acid. So 3 fatty acid plus glycerol in the presence of lipase. Starch in presence of amylase will give maltose, 2 molecules of maltose, or multiple, sorry. Starch is again a polysaccharide, multiple molecules of maltose will be formed. And maltose again, if given maltase, will form glucose. Yes. Protein will give protease, peptide plus polypeptides. Yes, that is formed or made together by peptide bonds, as we had discussed in proteins. Up till here, any doubt? Everyone should reply in the chat, everyone. We discussed about the definition, what is an enzyme, we saw its active properties, we saw the active site, what it depends upon, we saw the characteristic of enzyme, we saw the substrate, cofactor, and nomenclature. These things, any doubt anyone has? Everyone should reply, everyone. Okay. Fine, so we'll keep it till here. We'll move forward for classification and mode of action of enzymes in the next lecture, and we'll probably complete enzymes in the next lecture itself. Yes. Okay. Ayush, have you taken the attendance?
Yes, sir.
Okay, fine. So we'll keep it till here.